Hello, today I'm going to be discussing my 2CM4 design project. I did my project on using biocircuits for copper detection in living systems. Due to many natural factors such as global warming, industrialization, and man-made pollutants, environmental free iron copper concentrations have been rising over the past 2 decades. Through processes such as bioaccumulation and biomagnification, these copper ions can find their way into our diet. Copper is very hard to get rid of in the body and is toxic in high enough concentrations. It can cause nausea, diarrhea, and even death in extreme circumstances. In this project, we aim to optimize the design parameters needed to create biological circuit that is capable of notifying an end user if there are toxic copper concentrations in the medium of food or liquid that they are going to consume. How is this an optimization problem? Well, first let's go over how the biological circuit we are considering actually works. So a copper molecule is going to activate a promoter, which leads to transcription of a gene that produces a chromoprotein output. A chromoprotein is something that exhibits a brilliant pigmented color. So, in essence, copper concentration will lead to a bright dyed protein being produced, and the higher the concentration of copper, the more dye will be produced. Now, there are two things that makes this an optimization problem. One is the fact that we only want visible chromoprotein expression when the copper concentration in the medium is toxic to humans. This is something that can be done by altering the equilibrium value of the promoter, which is something that we can do in practice by selecting different promoter types that all serve the same function. We will define this as the lower bound of our circuit efficacy. So 2 comes after we've done 1, determining the upper bound of the copper efficacy. See, the thing is, copper is also toxic to the bacteria host that we're making these circuits in. So this means that although there is more chromoprotein being produced at higher copper concentration, there are also more bacteria dying. So there is a sweet spot between accelerating chromoprotein production and copper killing the cells in which we have produced these chromoproteins. And in this band, we'll have maximum chromoprotein produced. So at this production, we will define the upper bound of the circuit efficacy. So how do we come up with the equations to model this thing? Well, we need a few different equations for this circuit. In biology, there's something called the central dogma, which is essentially how all cells function and express genes. There's replication, transcription, and translation. We're going to need mathematical models for all of these processes. For the sake of time, I can't get into how to actually derive all these equations, but I go through it pretty in depth in the report. I'm just going to provide you with the final equations here. Since each cell produces chromoprotein and there are lots of cells in a sample, we can multiply the cell concentration by the chromoprotein concentration to get the total chromoprotein production, and this is a measure of how intense our dye will be. In my 2PO3 course, I actually built this circuit with an arbitrary promoter. Using the FEM section and the lab data that I obtained from that course, I'll create a numerical answer for what we deem visible chromoprotein production. We can determine the amount of cells that were in the samples initially by looking at the absorbance values for the control, and we can use literature to find constants for everything else. Note that these stay the same for the whole circuit even when we optimize them. The only thing that depends on the promoter is the equilibrium value, and in this lab we use a p-cusp promoter which has this equilibrium value. We can see there is just barely a color change visible at 0.5 nanomolars of copper in this lab, so we will input this value into FlexPE. We can then run the circuit for 10 minutes, as this is the time we ran it for in the lab. This gives us a numerical value for what we can use as the lower bound of our circuit. So we know that this much chromoprotein gives us a barely visible output. We now want to optimize our promoter to give us this output for concentrations harmful to humans. We import our flex code into Python, putting percent operators for both copper concentration and the K equilibrium values. Initially, we hold the copper values constant while changing the K equilibrium values. We optimize by using while loops that change the K equilibrium until the chromoprotein concentration passes the threshold value. We then do a second pass in smaller increments between the two values above and below the threshold to refine our constant, and a third pass to refine it even further. Here is a graph showing the 3 runs and the constant determined. We then use this constant to determine the upper bound of the effective circuit, starting by determining the order of magnitude that our solution lies on, then scanning through in one pass for an output. Using this process, this is the upper bound for the circuit efficacy. These answers mean that if we wanted to create a circuit capable of detecting copper toxicity in humans, we would have to have this equilibrium constant for a promoter, and it would give sensible results for this range of copper concentrations. Unfortunately, this model is limited because it does not take into account the fact that bacteria reach a stable colony after some time. It also doesn't take into account the fact that cells actively get rid of copper and cells may have chromoprotein after they die. However, we always need to make some assumptions when modeling, otherwise we could keep going about what we can include forever. The results from this project have pretty heavy implications because this may provide a new possibility to select iGEM parts for circuit construction and efficient mathematical modeling and optimization for genetic engineering circuits. Thank you for watching.